This video highlights our work to couple the functional structural plant model CPlantBox to the Unreal Engine in such a way that it elevates the workflow from a data production pipeline to a true model embedding. First, I will show what practically the coupling does. CPlantBox generates a plant structure consisting of connected segments in 3D space that are generated by a process-based model that encodes plant growth according to a rule set, determined through experimental measurements, measurement calibration, or functional modeling. This structure is expanded into a 3D geometry by its visualization module. This module creates basic shapes made of connected triangles around the segments to form the surface geometry. The Synovis framework transmits the geometry to the Unreal Engine, where a virtual world instance is calibrated to the environmental conditions, including measured sun radiation, cloud coverage, and similar. In Unreal, the light is measured at each segment and the resulting information is transmitted to CPlantBox, where the photosynthesis is simulated. The plant structure at the next time step is then generated. This is illustrated by the measurement in a model scene. A model scene means that we are embedding a certain aspect of the plant surface onto the Unreal Engine to evaluate light intensity at each predefined point, in this case the plant segments. The example I am showing is running on my local machine using an NVIDIA RTX 2060, so while the workflow is focused on supercomputers, it does run locally and evaluates the light intensities just as well. For illustration purposes, there are short pauses between each action. We transmit each plant organ geometry to the engine to later be able to disambiguate organs for texture mapping. The visualizer in CPlantBox produces full geometric information, including information about which triangle is produced by which segment of the graph structure. The measurement is done by virtual screen space, measuring the illumination on the surface at arbitrary points, which is restricted to segments on the Python side. As indicated by the red and blue boxes above and below the leaf surface, we are evaluating the normal in world space without assuming a specific structure, which means that leaf bending and wind reaction could be implemented in Unreal Engine without disturbing the function of the measurement. The PlantBox photosynthesis module is implemented based on the Farquhar-Loening model. The level of embedding resolution, meaning how well defined the conditions are, depends on the use case. In our approach, we replicated the Selhausen measurement of 2016 for winter wheat by assuming non-limiting water and nutrient supply. Generally, the carbon uptake of the plant in a model will be limited by the plant parameters, which describe the plant phenotype, or by the environmental variables that can cause resource limitation. The CPlantBox FSPM computes a whole plant approach to solve the equations. The combination of different limitations and their influence on the steady-state reactions can be seen in the article written by Giraud et al. Our scaling experiment in field partitioning is based on a central measurement area that is surrounded by a plant buffer. This means that we have a central area per compute node. Each plant instance is controlled by a seed which depends on its position, and this also includes buffer geometries. This means that in our scaling experiment, there's one central truth to the light evaluation. The workflow is as follows. The user defines a solid space that is distributed among the available computing nodes. CPlantBox initializes and updates the plants, weather, and environment in parallel at each time step. Each plant is assigned a position based on the total number of plants and the MPI field size within the corresponding compute node. To account for plant light competition at the boundaries of each computing node's field section, additional plants are included outside the area where photosynthesis is measured. These plants can either be simulated by the computing node itself or managed through communication with neighboring nodes. For the Selhausen experiment, we replicated sun exposure as well as the carbon uptake through the CPlantBox photosynthesis module. Our sun exposure variance is lower than the one measured in the experiment. We assume that some indirect lighting contributes to this effect. However, no additional indirect lighting was added to the simulation to limit the over-calibration of the model. The functional information that is produced by the FSPM can be utilized to generate synthetic data. We show this on the example of mapping leaf average light absorption relative to its capacity onto the plant geometry. Thank you for watching this video.